Hello, everybody, and thank you so much for the warm welcome. Before I get started, I wanted to share that I've been in a very fortunate position to receive various forms of funding from both industry sponsors and foundations that are non-for-profit. I hope those don't change in any way the way I look at scientific data. For the outline for today, I'll start by talking a little bit, very briefly, about chronic kidney disease and some aspects that I find important to point out. Then talk specifically about chronic kidney disease in cats, and go through the pathophysiology of progressive chronic kidney disease, and list very specific factors that I'm more interested in. And whenever applicable, I'll discuss briefly some strategies that we might want to use to start addressing those factors for progression in a clinical way. I want to highlight that when we talk about chronic kidney disease, what we're referring to is an abnormality in either kidney function or structure that has been present for at least 3 months. And so what that means is this is actually a very general term that applies to a heterogeneous group of disorders. In fact, we probably should be using the term chronic kidney diseases and not chronic kidney disease because they are very different. They have different etiologies, they have different histopathology, and they have different rates of progression. So we know that some diseases and some individuals will progress very quickly, and we look at progression based on changes in glomerular filtration rate over time. We also have some individuals that progress a little bit slower, as well as those that have progression that is accelerated by intermittent episodes of acute kidney injury. And so what we try to do clinically is to implement strategies that retard that progression of disease. And that's because we know that there's a self-perpetuation ability of kidney disease and renal injury. So essentially, you start by having a primary renal disease that takes down a few nephrons, and with that, reduces our overall glomerular filtration rate. Once that happens, you actually end up having adaptations that partially restore that glomerular filtration rate. However, those adaptations tend to become maladaptive and actually promote a further decrease in functioning nephrons. We also see accumulation of uremic toxins, which again promotes accelerated renal damage. So if you plotted glomerular filtration rate over time and looked at this, it might look like something similar to the graph on your right. So you see the primary disease having acute declines in glomerular filtration rate, some adaptations that make it a little bit better, but then once you have reached that progressive threshold, now the disease is going to start progressing rather quickly on its own. And what's interesting to us is what's happening towards that tail end. And what is happening is the development of tubulointerstitial fibrosis. That is the final common pathway for all chronic kidney diseases once you reach end-stage renal disease. At the cellular level, it's this cytokine milieu with a lot of names that we mentioned earlier, including transforming growth factor beta, which is a primary master of inducing fibrosis in chronic kidney disease. Now, cat people really like chronic kidney disease, you might know, and the reason why we really like talking about it is because it's very, very common. So it affects up to almost 4% of the general feline population, and depends a little bit on the studies, but almost up to 80% of cats that are 15 years or older, and you know that cat people have very old cats typically. We also know that renal disorders, air quotes, of which chronic kidney disease is the more common, are the leading cause of death for adult cats, adult and geriatric cats. So this is really important. In terms of etiology, there's many primary diseases that have been described in cats, but for most of them, we actually don't know the exact cause for it. We do know that on histology, most cats are affected in the tubular interstitial compartment, and so what we see is interstitial inflammation. We see tubular atrophy and fibrosis of the interstitium with associated secondary glomerulosclerosis. The rate of progression, however, is highly variable, and I'll show you one of the seminal papers that has evaluated this from the Royal Vet College group, and they've evaluated 213 cats with azotemic chronic kidney disease, creatinine greater than 2. They followed them for up to a year or up until they progressed, and they defined progression as a 25% increase in plasma creatinine concentration. What they saw was that almost half of the cats progressed within a year. That number was slightly lower if you looked at those in stage 2, and then slightly higher for those that were in stage 3 and 4. if you looked at how many of those cats reached stage 4, so the final CKD stage before death, then you'll see that a subset of cats in stage 2 and 3 ended up progressing through all of those stages. Now, if you're a cup half full type of person like I am, you can look at these data in another way, and you can see that actually a subset of cats remain in the exact same iris stage their whole life until they pass away of something else. Now, what were the risk factors for progression? There were 3 things which we'll refer to in this presentation that predicted progression. 2 of them for all cats. So hyperphosphatemia and proteinuria predicted progression for all cats. And then anemia was predictive of progression for cats in stage 2. So the question is, then what is promoting this ongoing inflammation and development of fibrosis in cats with CKD? And there are really beautiful, detailed reviews of the topic. Certainly in the constraints of this presentation, we can't be as detailed. So I'm going to focus on some of the factors that I'm more interested in. These will include single nephron adaptations to reduction of nephron mass, renin-angiotensin-aldosterone system activation, systemic arterial hypertension, hypertension, proteinuria, chronic kidney disease, mineral and bone disorder, accumulation of lipid in the renal parenchyma, hypoxia and oxidative stress. There's many other factors, notably aging, for example, and uremic toxin accumulation, which I won't cover today. So I highlighted this self-perpetuating nature of CKD, and so I want to start by talking about single nephron adaptations to renal mass reduction. And first, I'm going to bring something back from your hippocampus about glomerular filtration rate. So I want you to recall that glomerular filtration rate globally for the kidney equals the single nephron glomerular filtration rate, so what's happening in each nephron, times the number of functioning nephrons we have. And then, this is where I might lose some of you, so stay with me. GFR is determined by the filtration coefficient, so the coefficient, which is related to the surface area of the glomerular capillaries and the hydraulic permeability of the glomerular filtration barrier, and then the Starling forces in the glomerular capillaries, with the hydrostatic pressure in the glomerular capillary, which is this PGC, being the main force favoring filtration. So we know, it's been really detailed, what happens when you reduce the number of functioning nephrons in cats. So studies that have evaluated at surgically inducing nephrectomy in cats have compared what happens when you do a 3/4 nephrectomy relative to control cats, and they saw increases in single nephron GFR with associated increases in glomerular capillary hydrostatic pressure right here, so partial nephrectomy versus control, and then increases in ultrafiltration coefficient. So associated with that, we also had some morphometric changes that included some mild mesenterial expansion and increases in glomerular size. Hopefully you can appreciate that relative to the normal glomerulus, as well as areas of focal glomerulosclerosis. You can see here areas of glomerular scarring. So when we look at these single nephron adaptations and try to think about the pros and cons of that, if we look at the functional ones, we have preferential afferent arteriolar dilation with a consequent increase in intraglomerular pressure, which is really helpful in the acute setting because it's going to lead to an increase in that effective filtration pressure, right? So increasing in GFR. However, that also leads to hypertensive glomerular injury and then the development of glomerulosclerosis. So while it is helpful in the short term, in the long term it's unhelpful because you'll end up losing nephrons and consequently glomerular filtration rate. Same thing for that glomerular hypertrophy, right? It's helpful in one way because you increase your surface area, so you increase your filtration coefficient, and therefore you increase the single nephron GFR. However, as you have a disordered growth, again, you promote glomerulosclerosis and the loss of functioning nephrons. So these are really helpful in the short term, really unhelpful in the long term. The other adaptation we see is an overactivation of the renin-angiotensin-aldosterone system, which is associated with preferential constriction of our efferent arteriole. And if you remember, that's sort of the pressure relief valve of our glomeruli. So if you constrict it, again, you're going to have an increase in intraglomerular pressure. And you might recall the renin-angiotensin-aldosterone system, or RAS, in a very simple way, at least that's how I recalled it from vet school and even early in my residency, as this cascade that starts with angiotensinogen and then sequential cleavage by renin, the rate-limiting step, and then angiotensin-converting enzyme to generate angiotensin II, which is the main effector peptide of the system. Angiotensin II then directly and then indirectly through interaction with aldosterone is going to have or stimulation of aldosterone, I should say, is going to have a lot of effects that are going to be summarized as an increase in renal sodium and water reabsorption, and increases in systemic vascular resistance, stimulating antidiuretic hormone and norepinephrine secretion. So overall, what we have is an increase in extracellular fluid volume and blood pressure. This is really helpful in a lot of scenarios, including just maintaining normal blood pressure throughout our day-to-day, and clinically relevant scenarios like hypotension and bleeding. However, it's a really unhelpful activation when you have kidney disease. And that becomes problematic because it might promote systemic arterial hypertension, but also because of what I was referring to of promoting this intraglomerular pressure and hyperfiltration, which, as we've shared earlier, is going to be problematic. The RAS is also problematic in non-hemodynamic ways because both angiotensin II and aldosterone are actually directly pro-inflammatory and pro-fibrotic. They do these effects in several different ways, including inducing reactive oxygen species and increasing our expression of cytokines like transforming growth factor beta. So what do we know about the RAS in cats with chronic kidney disease? If you haven't noticed, cats like to be challenging in some ways, and their data is a little bit conflicting and somewhat mixed. In one hand, there's some data that suggests that plasma renin activity, which again would be sort of a really great marker of the circulating RAS since it's the rate-limiting step, it's either normal or actually low in cats with hypertensive CKD. So a lot of the studies have focused on hypertension in CKD. On the other hand, plasma aldosterone tends to be increased in these cats. Now, I would argue that having a normal plasma renin activity in a hypertensive animal would be inappropriate. So I would consider that as evidence that maybe the RAS is actually overstimulated. We also know that in some studies, including both spontaneous disease and chronic kidney disease models, plasma renin activity was also increased. So I think at least on average, there's data to suggest that at least in some cats, the RAS is overstimulated. Now, our expanded view of the RAS has continued to grow over the last few decades, and what I described earlier is now called the classical RAS pathway. We also are well aware that there is this other pathway called the alternative axis or alternative pathway that includes different angiotensin peptides and different receptors and is there as a built-in intrinsic counter-regulatory system that has actions that exactly oppose those of the classical system. So natriuresis, vasodilation, anti-inflammatory, anti-fibrotic pathways. Little work has evaluated that in cats with CKD. It is a really fascinating topic, and if you are excited about it as I am, then I highly recommend that you attend the DACVIM forum because we're going to have some abstract presentations as well as whole lectures on the RAS that include the expanded RAS in cats. Now, in addition to the circulating RAS, we also know that every tissue in the body also expresses their own local or tisular renin-angiotensin system. And that includes the kidney, where the concentrations of angiotensin II are several orders of magnitude higher than those in circulation. What we know about these systems is that the regulation of the local RASs is independent of that of the circulating RAS. We also know from work in both human beings and rodent models that the intrarenal RAS specifically seems to be of critical importance to the pathophysiology of chronic kidney disease and hypertension. There's a handful of studies that have looked at the intrarenal RAS in cats. The early studies did that through immunohistochemistry. So for example, this is one of the earlier studies you can see by the snapshot of the article, right, with the older, very pretty looking font, that has looked, for example, at the staining for renin in cats with CKD relative to controls and found that the RAS was overactivated, at least in a subset of those cats. There's another group that also did angiotensin II as well as renin IHC, and they found that some of these, including angiotensin II, the staining correlated with worse disease, specifically glomerulosclerosis in cats. Our group also evaluated the the intrarenal RAS in cats with kidney disease. We did it in a different way. So we collected very fresh post-mortem tissues from cats that were being unfortunately euthanized from chronic kidney disease. And we did RT-qPCR. So we looked at mRNA levels for several genes, including angiotensinogen, renin, angiotensin-converting enzyme, and the angiotensin II type 1 receptor. And what we saw was a significant increase in the transcription of angiotensinogen in cats with CKD relative to controls. That was very exciting to us because we know that the intrarenal RAS regulation appears to be related to the generation of angiotensinogen and not renin, so that was exciting. Interestingly, they had a downregulation of angiotensin-converting enzyme, which was unexpected. After we did all of the study, then continued reading more and wondered if perhaps other pathways other than ACE might be more important in the kidney, and unfortunately, we did not evaluate those in these studies. We also measured angiotensin I and angiotensin II peptides, and there was a significant increase in angiotensin I, but no statistically significant increase in angiotensin II. There was a numerical increase, it just didn't reach statistical significance. So I think because this is our study, I can more easily criticize it. I think the good thing about this study is that we used very quantitative methods. We looked at mRNA, which suggests that these things can be locally derived, right? And we evaluated the peptides with an LC-MS base. The bad thing is this was a really small study. Still, it took me almost 3 years to gather all of these cats. Also, the iris stages and blood pressures of these cats were not well known. This brings me to the next topic of systemic arterial hypertension. The pathway to get hypertension in animals with CKD is somewhat complicated, but I will highlight a few things. Associated with the loss of functioning nephrons, these cats or people or dogs might have endothelial dysfunction, increases in their RAS activity, sodium retention, and then sympathetic nervous system overactivity, all of which would promote hypertension. Now we know that there's this bidirectional relationship between hypertension and chronic kidney disease, with chronic kidney disease promoting hypertension and then hypertension promoting accelerated progression of CKD. We know that individuals with chronic kidney disease have increasing susceptibility to renal injury because they are less able to auto-regulate their renal blood flow. So again, hypertension is going to promote glomerular hypertension, glomerulosclerosis, and ongoing damage. What do we know about hypertension in CKD in cats? Again, cats are really fun, right? So in people and even dogs, a direct link between hypertension and survival is well documented. Cats like to make things a little bit interesting for us, right? So the available literature for cats would suggest that hypertension is highly linked to proteinuria, and then proteinuria is an independent risk factor for survival. I'll, however, highlight that in these studies, all of the hypertensive cats were being treated with antihypertensives, so it might be difficult to prove that hypertension had an impact on survival when you were treating hypertension itself. I am going to share something I didn't earlier about what happens with reduction of functioning nephrons, and that is an increase in urinary protein to creatinine ratio, which you can see here in this graph. So in cats with CKD, either the primary disease itself, and Dr. Hokamp is going to cover a lot of these diseases, or maladaptations to nephron loss, or hypertension, all of these can promote proteinuria. And proteinuria, although there is some debate on whether or not it is a marker or a mediator of renal damage, Most of the scientific community now would agree that it is actually also promoting direct renal damage through inducing, once again, several cytokines and growth factors and complement activation, which again would promote interstitial inflammation and tubular cell damage as well. So in cats, proteinuria is a very important predictor of survival and all-cause mortality, as well as CKD progression. However, and this is really interesting, in the one very high-level study, level 1 study, that has evaluated the use of benazepril, an angiotensin-converting enzyme inhibitor that has antiproteinuric effects in cats, and so these were cats that were randomized to receive either benazepril or placebo, benazepril did, in fact, lower proteinuria and improve the cat's appetite, but did not improve survival. Overall. Now, I think there's a few reasons that might explain this. On one hand, this was looking at all cats that were, some of them, not proteinuric. When they looked at the cats that had a urinary protein to creatinine ratio over than 1 specifically, then actually there seemed to be more of a difference, but there were only 13 of those. So, of course, this did not reach statistical significance. The other thing that I think is really interesting is we used benazepril, which is a drug we use a lot. There's some preclinical data out of the University of Georgia that would suggest that there might be better ways or more effective ways to block the RAS. And I'll show you these data. This is data from part of our group, and they have looked at the pressor response test, meaning cats were administered angiotensin 1, and then we observed an increase in systemic arterial pressure. These cats were under anesthesia for ethical purposes. And so when they were administered placebo before the pressure response test, they experienced a dramatic increase in blood pressure. That increase in blood pressure was well attenuated by benazepril. This is at 90 minutes after administration and 24 hours after administration, but even more so when cats were administered the angiotensin receptor blocker telmisartan. So these data suggest that maybe telmisartan might more completely block the renin-angiotensin-aldosterone system in cats. Now, there's been 2 very nice, large, randomized placebo-controlled studies that have looked at the effects of telmisartan on hypertension, one in in Europe and then another one in the United States. Both of them showed significant reductions in blood pressure relative to placebo over time. However, these studies did not look at progression or survival, so it's still to be unanswered whether that could be a benefit to these cats. Now, if we look at what can we do and what are the current guidelines, the International Renal Interest Society currently recommends using antihypertensive therapy with either amlodipine, the calcium channel blocker, or telmisartan, the angiotensin receptor blocker. I just shared those data with you. For proteinuria, they recommend using a RAS inhibitor, like an angiotensin-converting enzyme inhibitor or an angiotensin receptor blocker, and feeding a clinical renal diet. As most of you probably know, there's many features to renal diets, one of them being having a reduced protein content. And so the recommendation to reduce protein content in animals with proteinuria comes because high protein diets actually at the glomerular level are going to increase interglomerular pressure. That's because they preferentially dilate our afferent arteriole and constrict the efferent arteriole. So with that, you have a promotion of proteinuria, increases in, again, hyperfiltration, increases in the RAS, and worsening fibrosis. Now, there's some debate about the pros and cons of reducing protein in people with chronic kidney disease, with benefits being improving our glomerular hyperfiltration and proteinuria and uremic toxins, for example, and potential risks being energy wasting, specifically protein energy wasting and loss of lean body mass. So the overall recommendation really focuses on reducing protein while ensuring that lean body mass is preserved. Now, you better believe that if there's some debate for omnivores, there's definitely a debate for carnivores as well, right? And so it is, to some degree debated whether or not renal diets are indicated for cats with azotemic chronic kidney disease, with some of our colleagues believing that there's not a lot of, or at least not sufficient evidence to support their use, and some colleagues really focusing on the data that I'm about to show you that shows a survival benefit. I'll show you the data from 2 studies that have looked at survival in cats with chronic kidney disease that received a renal diet. In this first study, this was a really interesting design. So all of the cats that were diagnosed with azotemic chronic kidney disease were, or to the owners, it was recommended that they received a renal diet. For either cat or owner-related reasons, some accepted and some did not. And so then the investigators, again, of the Royal Vet College looked at how did these cats do over time. And what they saw was that the renal diet, which is that solid line, had improved survival relative to the no renal diet group. In this other study, cats were actually randomized to either receive a renal diet or a maintenance diet, and then followed over time. And what they found was that the cats on a renal diet had improvements in a lot of their functional markers, like BUN, for example. They also had a significantly better crisis-free interval, so they had fewer uremic crises. Actually, no cats in the renal diet had uremic crises relative to the maintenance diet. And they had an improved renal survival as well. Not all-cause survival, but renal survival was improved. So overall, this provides this support to most of our recommendations to feed cats a renal diet. Now, of course, there's several features of these diets, and one certainly is the modified protein content. The other one is modified phosphorus content, which is also really important. And this brings me to the next factor for progression, which is chronic kidney disease, mineral, and bone disorder. This really interesting, sort of newer term describes these collective changes in bone and mineral metabolism and alterations in calcium and phosphate homeostasis that happen with chronic kidney disease. These are really prevalent in people and animals with chronic kidney disease and seem to all stem from one thing, which is the renal inability to eliminate phosphate once you have a drastic reduction in glomerular filtration rate. So those changes in glomerular filtration rate and consequent renal retention of phosphate actually promote increases in fibroblast growth factor 23, which will then reduce 1-alpha hydroxylase activity, therefore reducing the conversion of vitamin D to its active form, calcitriol. Then the hyperphosphatemia and the low vitamin D together promote increases in parathyroid hormone. So we end up developing a renal secondary hyperparathyroidism. We know that chronic kidney disease, mineral, and bone disorder is associated with increased morbidity and mortality, again, across species, human and veterinary species as well. I mentioned earlier that hyperphosphatemia predicted progression in cats. I also mentioned that cats that were fed renal diets that are phosphate restricted did better than those that did not. So I think this is probably not up for debate at this point. One thing that we have been really interested in the last maybe couple of years or maybe a little bit longer is to what degree should we restrict phosphorus? And I'll show you some data that have specifically looked at that. And that's because of the challenges with the level of phosphate restriction and the dietary calcium to phosphate ratio. So in this first study that I will share, this was describing 19 research colony cats that had iris CKD stages 1 and 2 and were originally on a highly phosphate-restricted diet with a relatively high calcium to phosphorus ratio. In the beginning, in the first 6 months, these cats had improvements in their functional markers like creatinine and BUN and SDMA. However, over time, these cats developed both total and ionized hypercalcemia while being fed that highly phosphate-restricted diet. These cats were then transitioned to a more moderate phosphate-restricted diet with a lower calcium to phosphate ratio and ended up having a resolution of both their total and ionized hypercalcemia. There's also another study that describes the effect of sort of attenuating that phosphate restriction in cats and the effect on hypercalcemia. So in this study, the investigators, again from the Royal Vet College, looked at cats that were either already hypercalcemic at the diagnosis of CKD, or cats that developed hypercalcemia while eating a renal diet that was more phosphate restricted. And then in 10 of those cats, they actually transitioned their diet to a more moderate phosphate-restricted diet with a lower calcium to phosphorus ratio. And so what they saw, this dashed line is sort of the mixed model line for the cats that transition, and this is for the lines that were already hypercalcemic at baseline. You can see that when they transitioned to a renal diet, they had improvements in both their ionized and their total calcium. And so the conclusion is attenuating that level of phosphate restriction might help these cats not develop hypercalcemia, which is obviously problematic because we know that hypercalcemia predisposes these animals to dystrophic mineralization, including in the kidneys, which is not something that you would necessarily enjoy in an animal that already has chronic kidney disease. Now, in people, obesity and hyperlipidemia are independent risk factors for development of chronic kidney disease. And there's many reasons for that. One of the things that is postulated is that renal parenchymal lipid accumulation is actually going to have negative effects, again, through reactive oxygen species generation, and mitochondrial damage might promote ongoing inflammation and then develop fibrosis. While similar data are not available for cats, there's some interest in the interstitial lipid accumulation. This image here that I'm showing you is from a renal ischemia model in cats. So when investigators applied renal ischemia and then did histology 42 days later, hopefully you can appreciate here that this area where this would be a tubule that got damaged, and here you see that the tubular membrane got disrupted, so we call that tubulorexis, and lipid that would have been inside of the tubular cells was leaked onto the interstitium. And associated with that, then you have a granulomatous inflammatory response. This same group out of UGA published a really nice review of chronic kidney disease in aged cats that I highly recommend that you read if you're interested in the topic. And they comment that interstitial lipid is accompanied by granulomatous inflammation very commonly in cats in iris stages 2 and 4. Now, there's this one study that has looked specifically at renal interstitial lipid accumulation in cats with chronic kidney disease. And they showed that renal lipid was present in actually all of the cats that they evaluated. And you can see it here highlighted with osmium tetroxide because it really brings that dark color and helps you see the renal lipid. Now, our own Dr. Quimby has published a really interesting study that looked at the frequency of histologic lesions in the kidneys of cats that did not have functional evidence of chronic kidney disease. So these cats had normal markers of renal function. And they've described a lot of things, but one of them was interstitial and tubular lipid. So here in this graph, we have tubular lipid and over the ages of the cats, younger to older. And this is interstitial lipid. And it's really interesting to see that the cats over 15, all of them had higher, or at least some interstitial lipid being present, and increases in tubular lipid as well. And associated with that, hopefully you can appreciate here, was always that granulomatous inflammation. So the question is, could there be a role of lipid in the development of CKD in cats? If CKD is more common in aged cats and lipid is more common in aged cats, might this be one of the reasons for that? Which I think is a really interesting thought experiment, especially knowing what we know after experimental kidney damage. Now, the last factor I will talk about is renal hypoxia. You might recall that even in the best of days, the kidneys are particularly prone to hypoxia. And that's because they have a really interesting vascular arrangement that makes it so the PaO2 in health in the cortex is pretty good, but as you go down into the medulla, it reaches very hypoxic levels. So there are areas of the kidneys, for example, the straight proximal tubule, that are both simultaneously very, very active and working under relatively hypoxic conditions. So even the healthy kidney is predisposed to hypoxia. Now, there's a number of challenges to oxygenation in CKD. Those might include some systemic factors, so for example, hypotension, dehydration with decreased renal perfusion, as well as anemia of chronic kidney disease. These are easily modifiable factors, but certainly things that we might see that promote renal hypoxia. We also have an imbalance in vasoactive molecules. So for example, RAS activation would be a typical example that promotes arterial or vasoconstriction and therefore decreases the blood flow through the peritubular capillaries and therefore promoting a hypoxic environment. And the other, which I think is one of my favorite things to consider, is how fibrosis and the inflammatory infiltrate actually widens the distance between the peritubular capillaries and the blood supplies, and the tubules themselves, and therefore increases the oxygen diffusion distance. And so therefore promotes hypoxia as well. So there's this thing called the chronic hypoxia hypothesis, which is sort of this unifying theory to explain progression of many chronic kidney diseases. And the thought is that chronic hypoxia then promotes oxidative stress, activation of pro-inflammatory and pro-fibrotic pathways, and immune cell infiltration matrix deposition, therefore promoting tubular interstitial fibrosis. And then the fibrosis disrupts your peritubular capillaries, increases oxygen diffusion distance, therefore promoting hypoxia. So we're stuck in this sort of vicious cycle in here. Now, cellular adaptations to hypoxia are governed by something called the hypoxia-inducible factor family. And in this family, one of the very important factors is hypoxia-inducible factor 1 alpha. In the conditions of hypoxia, hypoxia-inducible factor 1 alpha is stabilized with the beta subunit, goes into the nucleus, activates the transcription of numerous genes, most of which are associated with angiogenesis, erythropoiesis, so erythropoietin would be one of the genes that would be activated by HIF, as well as cell proliferation and survival. Now, you probably already know where I'm going with this, right? So helpful in many aspects, but not in all aspects. When HIF1 alpha to other HIFs ratio changes favoring HIF1 alpha, it actually increases NADPH oxidases and reactive oxygen species, again generating oxidative stress, inflammation, and fibrosis. And in fact, HIF expression is correlated with worsening of disease in several rodent models and people as well. Our group did look at transcription of HIF1 alpha, as well as several other mediators of fibrosis in cats with naturally occurring disease. These were the same tissues that we did our RAS study on. And what we found was that there was a significant increase in the expression or transcription of HIF1 alpha in CKD kidneys, as well as transcription of transforming growth factor beta in CKD kidneys relative to controls. We had also documented that in tissues from cats that had undergone experimental renal ischemia, transcription of HIF1 alpha was associated with worse scores of fibrosis. So the higher the transcription of HIF, the worse fibrosis scores. Now, certainly these data just show association, no causation whatsoever, but at least it shows us that hypoxia is a specific challenge that cats with CKD are facing. So in conclusion, chronic kidney disease is a really relevant cause for morbidity and mortality in cats. I'm going to quote my husband, who's a poultry veterinarian, often uses this term that I love, and say that CKD progression is multifactorial and poorly understood, right? And so because of that, really strategies that we use to slow the progression should be multimodal because there's not one thing that is progressing the disease. And then I will say that unfortunately, many of you know this clinically, a subset of cats currently is going to progress no matter what you do, and so really we should continue studying the factors for progression so we can offer better alternatives. With this, I'm really grateful to you for your attention. I'll conclude this presentation. If there's time for questions, I'd be happy to take some.